Well, why don't I just jump into things then? So, thank you both for being here. I'm super excited about this. I've been talking to Mike for a long time about how I think there's a lot of really important connections between the work he does and the interoception allostasis ideas that Lisa Feldman Barrett and her colleagues have been putting out. So, I'm really excited to have this come together. I'm hoping both of you can do a presentation, then we'll have a little bit of time left over for discussion. Jordan and Eli, could each of you just introduce yourselves, and then we'll start with Jordan's presentation and then do Eli, and we'll take it from there.
Sure, sounds good. So I'll go first. I'm Jordan Theriault. I'm assistant professor at Northeastern. I've been working with Lisa and Karen Quigley for like 8 years now. So probably since 2017, I came on as a postdoc. Originally did a background in moral psychology and sort of social neuroscience. Transitioned out of that, got much more interested in the sort of constructivist and like biologically grounded perspective that Lisa and Karen have been running. Um, Initially was really interested. I think Ben, you'd cited some of the stuff that I had on social pressure. So I got super interested in sort of thinking about how social pressure might work from like a biological and allostatic perspective. So literally how others' expectations compel some sort of affective motivation driven by interoceptive signals in others. I can talk about that. That's not so much in the presentation I've got here, but the stuff that I got more interested in and that I've got a faculty position on now is Lisa really pushed me to think more about the underlying metabolism, like underlying metabolism as a sort of governing force for motivation. And so I got super interested in some particular problems in brain glucose metabolism that are underlying the bold fMRI signal. So my goal kind of right now is to think about how to detach the bold signal as just another measure of activity that people kind of lump in with like spiking, with the EEG, all of this stuff all together, and how to look at the bold as a real sort of measure of interest in itself. Largely to do with potentially like a like waste clearance and like local pH homeostasis. So that's kind of where I'm at now. And at the talk that I've got is kind of a broad overview of how the lab thinks about some of these topics and a bit about the metabolism stuff at the end.
Great. Fantastic. Thank you for that. Eli?
Hi, I'm Eli. I did my PhD sort of dual posted between the IASL where Jordan was and Jan Willem van de Meent's deep probabilistic programming lab. So I sort of stretched between the computational side of machine learning and the theoretical side of neuroscience. Then I did a postdoc with with Andre Bastos down in Nashville, where I sort of the looked at a lot of the implementation of predictive processing in the visual hierarchy, and of course, totally derailed my early career by, finding that the theory we all went in with was actually wrong, which is, what Popper says you're supposed to do in science, but everyone knows that's not actually how you do it. So, whoopsies.
It happens.
Yeah.
Fantastic. So thank you both very much for that. Jordan, could you just jump into things then and share your presentation with us?
Yeah, sure. 1 second. Let me get this set up here. All right. Oops. All right. You see this okay?
Yes.
Okay, perfect. So these are, feel free to stop me in any of this here too, but basically what these are is these are some slides from a presentation that Lisa and I put together last year for a sort of group talk in Paris on scientific concepts or concepts in neuroscience. It was a group of gathering of philosophers and neuroscientists in Paris to sort of cover some of this stuff. So let's jump into it. So first, yeah, I just wanted to point out here too, that this is all a team effort. So I think Lisa gets a lot of the attention for a lot of this, but we really try to make sure that people know the lab itself is run as a team. The way that we used to divide things up, or the way Lisa and Karen used to divide things up, is they'd say that Lisa handles everything above the neck and Karen handles everything below it. Um, which for me, coming in as a third director in the lab, it's like, well, where do I fit in? But I think looking at metabolism and sort of underlying systems perspective to sort of join those two halves and think about the bigger picture of it has been interesting for me. And I think metabolism crosses those boundaries a little bit too. So there's kind of room for everybody here. All right, cool. So basically, the way this is structured is just to talk about a couple of scientific principles that have organized the team's thinking here. And the first principle that I want to start with here is that we've tried to think about a bit differently than other labs, to think about the brain as a whole organ and to think about what, from that perspective, the brain as an organ's most important job is. We were really inspired by some work from Peter Sterling and Simon Laughlin. They have a book called Principles of Neural Design, which is, if you ever see Karen's copies, just earmarked to hell with like different footnotes and sticky notes in it. And so, inspired by that, right, we really want to have an explanation here that spans from like rats, humans, nematode worms. And from that perspective, the idea is that, uh, rusty on my animations here. The perspective is that we think the brain's important job is not to think, right? It's not thinking. Um, the most important job from our perspective is to coordinate and regulate tissues and organs of the body. Um, Considering that, we've got over 600 muscles, we're balancing dozens of hormones coursing through your blood. We're just pumping at a rate of 4 to 5 liters per minute. It's directing the GI system as it digests food. It's coordinating kidneys and livers, they excrete waste, coordinating your immune system to fight illness. And what we do is collectively call that whole process of regulation altogether allostasis. And so the other reason why we use this term allostasis is to draw somewhat of a distinction. So we want to say that allostatic regulation is about specifically energy regulation and efficiency of energy regulation at different levels of output. And we want to contrast that with the term like homeostasis, which is about regulating toward a set point, right? So the idea is that within allostasis, there's not a permanent set point. Um, there's a flexible sort of whole system change that has to adapt to perturbations. The other distinction as well is that we think of allostatic regulation as specifically predictive. So it's about avoiding errors as they come up and ideally leveraging other parts of the system to avoid having to have an error-driven correction. And from a homeostatic perspective, we think of regulation as reactive. So again, there's obviously individual systems in the brain and body that are working from a homeostatic perspective, but the idea is that at a collective, broad level, things need to be coordinated and regulated from these broader allostatic efforts of the brain. Um, so again, this is all drawing a lot on Sterling and Laughlin's work and this sort of cross-species analysis of requirements and design principles of a brain. And a really nice pull quote from Sterling and Laughlin stuff here is to say that the core task of all brains is to regulate the organism's internal milieu by anticipating needs and preparing to satisfy them before they arise. That's like a nice summary statement of what it is that we mean by allostasis when we're talking about it. And so what that means is that as the brain's regulating and coordinating the systems of the body, it's also modeling the sensory consequences of those changes from the body, and that's called interoception. So again, for us, interoception is the brain's modeling of sensory changes that are resulting from allostasis. And it's supporting allostasis in the same way that somatosensation is supporting feedback about skeletal motor control. So the idea is that your brain's always regulating your body. So it's always modeling the body's sensory state. This is something that's always on. And it's on, we make this point a lot, even when you're lying perfectly still in a resting state fMRI. So a lot of people have a very cognitive focus when they're talking about resting state fMRI. They'll say, it must be modeling internal thought or self-directed thought or mind wandering. And we really kind of make the point a lot that there's a lot of stuff that has to be going on all the time, especially as it regards bodily regulation. So you can't have these sort of cognitive blinders on and think that it all has to do with thinking. And that's this mistake of thinking about whether the brain is for thinking or whether it's for allostasis. So just an example thing of how we've tried to study this. So we had a resting state connectivity analyses that had tried to identify brain architecture responsible for coordinating and regulating the body. So in this work, this was from 2017, and we just had a replication at 7 Tesla out in Nature Neuro that's in press right now. Um, so first, what was done was the lab had found track tracing studies of macaques and other mammals to identify cortical areas that have direct projections to brainstem nuclei that are involved in regulating the autonomic nervous system, immune system, and endocrine system. So in other words, what they were looking for was cortical areas that were important for performing allostasis, proven by direct projections. Then what they did is they identified the homologous voxels in the human brain, used those as seed regions, and then they looked at resting state connectivity, oops, between each of those seed regions and the rest of the brain, where each one of those seed regions would produce one discovery map. What they did then was perform cluster analysis on those discovery maps to find clusters within each of them. And then they identified where they would have overlap at a broad level or across all of the discovery maps to find points of convergence across all of these different discovery maps from seed regions. And when they did that, what they'd find was two distinct networks and some overlapping regions between them that seem to be critical for allostasis from these projections. And so again, they'd replicated this in some HCP data, and then more recently in a large 7 Tesla sample that we have as well too. And what was done was that analysis that basically produced one integrated system that was comprised of two brain networks, which you can recognize here as looking quite a bit like the default mode network on the left and the salience network on the right. And those would overlap in a set of hubs, some of which are members of the Rich Club hub, which are thought to be these backbones for communication across the brain. One thing pointed out here, I had this in the original talk, and it's just worth flagging again, is that there's a lot of work that's been looking at gradients across the brain. So Daniel Margulis was someone who'd organized this conference in Paris. He'd been looking at these gradients from sensory surface to higher level abstraction, from sort of default mode to sensory surface. He was showing that he basically would interpret that as saying that there's sensory surfaces on one end, and then there's the default mode network on the other for abstraction. So we'd think of a gradient in the brain between exteroceptive sensation, external sensation, and abstraction. We're finding the exact same networks here, so it's consistent with that. But from our perspective, what we'd say is that those core networks aren't necessarily associated with abstraction. They're related to a different sensory surface, which is that interoceptive sensory surface. So what you have is still a gradient running from exteroceptive sensation all the way to the interoceptive surface. But again, other people, when you have these sort of cognitive blinkers on, will interpret that interoceptive surface as being about abstraction instead of being about visceral regulation. And again, we've got a bunch of stuff from our group that's sort of making that point here. All right, so again, summing up, first principle here is just to say that your brain's regulation of your body is at the core of brain structure and function. And so therefore, what we'd say is that it's at the core of your mind. There's a core aspect organizing cognition that comes from this allostatic perspective. Then what we'd say, right, is that your brain's most important job is not for thinking. It's not feeling, it's not seeing or hearing. What we'd say your brain's most important job is to run your complicated body. And again, once that animation stops here, we have a paper that just came out of this too that was just really trying to drill down and make this point. So this perspective just came out in Neuron. We are saying it's not the thought that counts, allostasis at the core of brain function. And if you want the sort of most up-to-date version of all of these arguments that are here, that's the one that we just put out. We tried to synthesize a lot of that anatomical and functional connectivity work into that paper. All right, so that's point one. So point two is to talk a little bit more about not just talking about where allostasis might happen in the brain, but how specifically the brain achieves it, right? So the idea here is, again, that allostasis is about predictive regulation of the body. And the most efficient way to regulate a body is to predict the body's needs in advance and to correct those predictions when necessary. So the claim that we want to make here is that the predictions in the brain are organized, predictions in the brain are organizing concepts in the mind. So these are a whole bunch of papers. The point here is just to show that there's a whole broad and really growing literature tradition that's emerged in the decade, focused on understanding how the brain predicts. So predictive coding, free energy, all of this stuff, I'm sure you guys are familiar. There's hundreds and hundreds or thousands of papers on this stuff that's providing evidence for predictive functioning in the brain. What I'm going to do here is just talk about the gist of this, but with their lab's own particular theoretical twist on it. So the general idea is this, which is that your actions and experiences are beginning as memories, and those memories are similar to the present in some way. And so you're not necessarily aware of yourself remembering, but this is what's happening. It's what's happening. We have a prior experience that's projecting onto current experience. The idea is that your brain is re-implementing signal ensembles from the past that are similar in some way to the present. So in psychology, when something is similar, when things are similar to one another, we'd call them a category. So prediction signals from this perspective are categories. When the brain's predicting, it's constructing a category of possible futures. And so these predictions are continuously being tested against the physical signals that are streaming into the brain from your body and from your sensory surfaces as they're sampling that outside world. So to see how this works, you can think about the last time that you were thirsty and when you drank a glass of water. So within seconds after draining the last drops of that water, you probably felt a lot less thirsty. But the thing that's weird is that it actually takes about 20 minutes for that water to reach your bloodstream and for the associated signals to reach your brain. So what is it that's relieving your thirst 20 minutes earlier than when those signals reach the brain? The argument is it's prediction, right? It's anticipation of those allostatic and interoceptive consequences. And so again, over the course of your lifetime, your brain's had thousands of opportunities to learn that certain motor commands involved in drinking, along with the feel and the taste of water in your mouth, are eventually going to quench your thirst. Another example here that I really like, I don't know, have either of you guys seen this example before, or do you recognize this picture?
I don't.
Cool. So this one, Lisa's used this a bunch of times. So the example here is that if you look at this image, it's difficult to make out what this is, right? So your brain's searching through a whole lifetime of past experiences, issuing thousands of guesses, weighing probabilities, trying to answer the question of what is this thing most like? Uh, and the point here, right, that we really want to emphasize is it's not asking, what is this? It's asking, what is this thing most like? It's trying to draw on memory, past experience to put this ambiguous sense data into a category. And so if you're still seeing only black and white blobs, and the point is that your brain hasn't found a good set of predictions yet, and you're in what we call a state of experiential blindness. And so I can cure your blindness. Oops, if the animation works, there we go. So if you haven't figured it out, this is a bee. You can see its head here. You can see the tail, a wing over here, tail over here, leg, and then we'll put it back. And so it should be possible to see these blobs as a bee now, right? Pretty reasonably. And the point is that what your brain is doing is it's creating new predictions thanks to the color photograph that I showed you. So what those predictions are doing is they're changing the way that you experience the blobs, the ambiguous sense data. And what we've done is we've constructed a prior experience, we've used a prior experience to help your brain construct the image of a bee so that you see a bee even though there's not really a bee present on the screen, right? It's not what's on the screen here that's changed. You've changed, your prior experiences have changed, and they've helped you construct this experience. So the point is that the whole process, which is making meaning of ambiguous sense data using past experience, is responsible for all the experiences that you've had in your life and the actions that you take. So the claim is that the same process of meaning making here is how psychological events, like instances of emotion, when we talk about construction of emotion, are being made as well too. They're assembling ambiguous sense data, a lot of it from the body in the case of emotion, to categorize it into a particular experience. So again, zooming out a little bit here, the idea is that physical signals are constantly streaming into the brain. On their own, those physical signals don't have inherent meaning or inherent psychological meaning, just like you experienced with the blobby image here. The point is that the physical signals become meaningful as they meet the prediction signals that are already present in your brain. And if those signals match, right, then the incoming signals are going to be categorized and explained. And if they don't match, then what we would do is call that a prediction error, right? So your brain encounters a prediction error. It can adjust what it predicts next. And we have a fancy change for that term, being funny here. There's a term for when, your brain experiences something, changes what it's going to predict next. We call that learning, right? So the point here, right, is that category construction is really about meaning making. So what your brain is doing is it's starting off with a category of possible futures, and then it's ending with specific instances of actions and mental features. So this is what we're saying is the basic operating principle of your brain. And in fact, when taking it from this perspective, right, a lot of the major topics that as psychologists would normally be interested in fall under this more general umbrella of meaning making when it's being considered in this sense. So again, predictive processing, interpreted with this particular spin that we have on it as category construction, is uniting a lot of these different folk categories into one common framework. Uh, and it's a common framework that has efficient energy regulation really at the core of it as well, too, from an allostatic perspective. So again, the idea here, just to summarize the second principle, and then I'll try to get quickly through the third one so that Eli's got some time here. Is just to say that your brain's constructing your experiences. So what you're seeing, what you're hearing, what you're feeling, it's constructing those as it's predictively controlling your body. And so the concepts that we're using, even the concepts that we're using as scientists, are ultimately stemming from how your brain has clustered up the physical signals that it's received in the world and how it's chunked those up into categories to help facilitate meaning making. And so again, that is predictive processing, but it's predictive processing that's interpreted through a constructivist lens. All right, so the last principle, which is more of the work that I've focused on since working with Lisa and starting up here, is that none of this stuff comes for free. So encoding all of these physical signals in the brain as prediction error has to have some metabolic costs that's attached to it. And I've spent a couple of years now trying to disentangle some of the research related to those costs. So again, as an entry point into this and where I'm at with it right now, I'm just sort of focusing on the bold signal, so the blood oxygen level dependent signal. And again, when we're talking about observing activity in fMRI, um, this is almost always what we mean, right? So what we're doing is we're showing people a stimulus. Um, we're seeing a bold increase in associated regions like visual cortex. Um, but what I want to push on here, right, is what really is activity, right? Activity is kind of a clunky term. It's combining a lot of, it's just basically saying that something's happened. And I'm not the only one who said this. In 2008, Nikos Logothetis, as well as this other paper by Singh, had really been urging a lot of caution in how we interpret bold signal. This matters, right, because it was Nikos Logothetis' work that had really established this link from the bold signal itself down into local field potentials, and then from there to local synaptic activity. And so I'm using dotted lines here for correlational and solid for causal, but the point is that these linked sort of helped settle for a lot of people this interpretation that the bold is an index of local synaptic activity, and a lot of people pretty much just left it at that. So that we could go on doing a lot of fMRI experiments and thinking we've got this index deep down into synaptic activity. But it's more complicated than that, right? So again, you guys probably know this, but the bold signal itself, right, is reflecting these changes in local blood oxygenation. And you get a bold signal in the first place because active regions are going to increase blood flow more than they increase oxygen use. But what's unusual and what I've been interested in is that something else also happens during that same process, which is that local glucose metabolism actually increases quite substantially as well, at the same time as you get everything else happening here. So to understand why that matters, this probably is totally unnecessary for you guys, but just quick second refresher. Most cellular energy is being produced by metabolizing glucose and oxygen together. So in this case, for fully oxidative metabolism, you get about 32 ATP per glucose. At rest, that is what's happening across most of the brain. You get a coupled ratio of oxygen to glucose of about 6 oxygen per 1 glucose consumed, It's fully oxidative. But again, in these active brain regions or in these stimulus elicited patterns of bold activity that we're seeing, we get something different happening, which is we get anaerobic metabolism. We get active regions using glucose without oxygen, which again gets us about 2 ATP per glucose. It also generates free hydrogen and lactate. Um, so what we're getting is that this, what we normally think of as activity is in fact, right, it's a couple of different things, only two of which here, the blood flow and the oxygen use gets us the bold signal, but we're also getting this combined increase in anaerobic metabolism. And so when we talk about activity, we need to actually consider everything that's happening locally as well. I can pull up some data on this too if you guys are interested, but I'm just going to skim through this for the second. So again, the point here is, why does this matter? It matters, right? Because anaerobic metabolism, if you exercise at all, means lactic acid buildup, or it means lactate and free hydrogen. So in other words, what's happening is these active brain regions are shifting their metabolism in a way that could potentially acidify themselves, which matters because neural function is sensitive to pH. As regional acidity increases, firing rates will decline. So fortunately, though, that increase in blood flow that we get, that we all knew was there for the bold signal, can potentially clear that acid out. And because that blood flow is increasing more than the smaller increase in oxygen metabolism, what we get is a bold signal. And so if we put all of that together, what you can get is this causal pathway where we think about having this cascade from anaerobic metabolism to lactic acid to blood flow to clear it to bold signal. You have a potential problem of local pH happening, which the blood flow then helps to regulate. And so again, I'm not the only one who's proposed this. Denuzzo et al. Had actually mapped this out and done Denuzzo and Doug Rothman and people at Yale Medical School had done some really, really great stoichiometry work and models to map this out and make this argument. But what we can do too is go a little bit further and ask what is the specific neural activity that might be driving this cascade? And so again, to get at that, we can superimpose the Logothetis model from 2001. We can update it since we know a bit more now than we did in 2001. So first, the Logothetis paper had emphasized this correlation between local field potential and bold signal. But what we know now more is that it's specifically with high frequency gamma band oscillations that the bold signal seems to be most correlated with. Second point is that those gamma oscillations are caused by interactions between pyramidal neurons and parvalbumin interneurons. I'm just going to focus on the parvalbumin here for now. But what's most interesting is that those parvalbumin interneurons, spiking in them seems to be glucose dependent. So there's been some studies run that have been substituted lactate or pyruvate or other fuels. And it seems that if you don't have glucose specifically, you'll get a suppression of firing rate. You'll get suppression of normal spiking within these parvalbumin populations. And so what that means is that normal oxidative metabolism isn't going to work as a substitute. There seems to be this glycolytic component that's really necessary here. And so in our 2023 paper, which is a big review of all of this stuff in a ton of depth, we'd hypothesized that those parvalbumin interneurons are driving this anaerobic cascade that's ultimately producing the bold signal. And so again, the critical part here is that all of these links can give us some insight into function, because as well, right, there's been some evidence that started to accumulate to link these gamma band oscillations over to prediction error encoding as well too. So some of the other work that I'm doing, which again, don't have time to get into, is I really think that there's been a bit of a people have gone a ways in thinking about their fMRI signals in a very stimulus-focused way. And I think there's a way of reinterpreting the bold signal as really a response to prediction error encoding rather than to just the stimulus encoding. So again, it's a lot to digest here. What I'm really trying to do is to lay a sort of spine through this research that we can connect other observations onto. This is obviously isn't complete, and there's links to sort of complete here or other observations to connect in. But the point is to just give a sort of big picture overview with this. And so again, to connect this back up to the big picture and then to try to wrap here, As we talked about before, what we were saying is that your brain performs two major functions. So in category construction, what the brain is doing is it's using memories to categorize physical signals in the present. And when it's learning, it's updating those categories. And another way to understand this is to say that there's two major directions that signals are flowing in the brain. So for learning, signals are flowing bottom up as prediction error. And if our model is right here, then the idea is that the prediction error signals have some quirks. So for one, we think that they might be using glucose to make less energy. They might be less efficient. They might be anaerobic. And second, if they're acidifying the regions that they're passing through, and blood has to flush these regions to regulate pH. Finally, this whole metabolic cascade of learning and prediction error encoding is potentially visible through the bold signal that we can actually measure easily in fMRI in the first place. So there's a particular type of signaling here that has particular regulatory responses to flush out waste lactate, which makes it visible to us as an fMRI signal. On the other side, right, within category construction, signals are flowing top down as predictions. And here, as I'd said, and if we think of the resting state as a case where people are not being bombarded with prediction error, where they're in a predictable environment, glucose metabolism in that case is energy efficient. It's largely oxidative. Because it's oxidative, it's not creating lactic acid that has to be flushed. And because it's not creating lactic acid that needs to be flushed, this type of top-down signaling or category construction signaling we'd suggest might be invisible to bold. So even within resting state fMRI, the fluctuations that we're getting might not be necessary. They're from many, many different signals. And I think it's a complicated thing to work out what it's all from in resting state. But the point is that the clear hemodynamic response, stimulus elicited response that you're getting, that's the bread and butter of task-based fMRI, might be indicating a very particular bottom-up process and missing a lot of this top-down signaling, which is really the bulk of the metabolic cost of the brain. If the brain's about 20% of the whole body metabolic cost in humans. So again, summing it up here, if we're right, and we're really, we're at the early stages of this, right? Then the idea is that prediction error is something that's supported by what's really a kind of unusual type of metabolism. And at the same time, prediction might be supported by a much more typical metabolic pathway, which is creating more energy, isn't creating the acidic waste that needs to be recirculated. And in the resting state, this is almost exclusively the type of metabolism that's being used. This has some psychological implications as well, because it's implying that the brain is metabolically optimized for category construction, for being in these predictable environments. And by contrast, it's suggesting that learning actually has a pretty big cost. If you're learning something well, then you don't need to learn it all over again. It's also, and this is what I meant by the experimental design, it's worth considering, right, whether the sort of randomized experimental designs that we use, especially in fMRI, are overemphasizing prediction error, and whether they're putting the brain into a situation that they're optimized to avoid. So the idea here is just that if your brain evolved to deal with environments where stimuli aren't popping suddenly into existence at the center of your field of view like this, then if you found yourself in an environment like that, you'd expect it to be pretty metabolically taxing on the brain because it's putting outside of the evolutionary zone that it's evolved to deal with. And you'd expect people to want to get out of that situation or to have to implement in regulatory strategies in the brain to try to deal with that very, very outside of the naturalistic norm perturbation. All right, I'm going to skip over this and just jump to the conclusion here. So if this is all true, right, if this model's right, then what do we do with this metaphor of bold activity that we'd started this section with here, right? So I think we need to give up on this idea that the bold is measuring generic function or generic processing. And instead, I think we can make a lot more progress by recognizing that the bold signal is coming from a very particular metabolic cascade that's driven by a particular neural populations, which might create the need for a particular regulatory response. And the argument is that bold is a measure of that regulatory response. All right, so altogether, that's just to give you some perspective on where we're coming from in the lab. Just summing up one last time, the sort of three principles here that are performing the work. First was this principle that the brain's core role is to regulate the body. The second principle is that concepts are reflecting how the brain groups and organizes physical signals to allostatically regulate the body. And then finally, the point is that even the simple act of encoding those physical signals is creating metabolic costs and consequences that need to be regulated. And if we want to connect the mind up to the brain, then we need to understand the underlying biological systems. So principles like this are just kind of general guideposts to help keep us oriented as we're working out a lot of those details. Cool. Thank you, guys.
Thank you so much. I really appreciated that. That's exactly what I was hoping for. And I love the dollar
Awesome.
signs. Awesome. Cool. Let's have Eli do his presentation. And Eli, don't worry about time for discussion. We'll just get through your presentation. I'll be following up anyway. So my main goal is to just make sure that Mike is familiar with interoception allostasis on some level. And then I'll be following up later anyway.
Sure, okay. So I'm gonna share my screen and I do wish to, sort of preemptively apologize. These are slides that I have rapidly recently updated to contain recent work. And other than that, they're somewhat old. Nonetheless, so yes, continue to share this window. So basically, the perspective that I sort of came to in graduate school, which is, I think if I was in private with Jordan, Lisa, and Karen, I would call it a nuance on what Jordan has just described. And perhaps in public, I might call it a difference of opinion, actually, which is that I really think, when we talk about the brain being an organ of regulation of the body, that actually means that its core job is not categorization, it's control. Now, if you have sufficient recording resolution such that you can treat, every spiking pattern like as a different quote unquote category, or treat, spike versus sub-threshold activity as a category boundary, then it's categorization, right? But like, anyway, so, right, see, this is the artifactual part. Okay, good. Yes, so I've actually sort of been working backwards through computational modeling of the of brain evolution in a certain way. So when I was in grad school, I did start from, this predictive coding or predictive processing as my sort of known factor that other people had, collected experimental evidence for. Nowadays I would have to be a little more nuanced on that because, as Jordan showed, prediction error encoding seems to be linked to synaptic activity and gamma band local field potential oscillations rather than whole circuit spiking activity. Nonetheless, we can sort of look at the neurophysiology of, shared views of prediction and control in cortex. Think about the ideomotor principle that's well established in psychology and start developing equations from there. This basically gave me like a couple of little modeling papers where I say, let's use log probabilities as a common currency. There will be such a thing as an not optical, optimal dynamics and a way of telling, what's your approximation error to how you should act. You can sort of unify with the math of predictive processing all over again while making sure that you track your regulatory targets. And, likewise, this is really how allostasis would map onto that control systems view of the brain. Interoception provides your sensors. Visceromotor and somatomotor action provide your effectors. And you have an internal model in the forebrain that basically helps you run a controller. Now, then there's the question that I left grad school with, and by which I'm going to sort of bridge to more recent work, which is if you're doing this sort of control as a process of bringing about pre-specified sensations, then where do those pre-specified, interoceptive sensations come from? Why are some such interoceptive sensations specified through development and learning and yes, evolution to be the ones that you regulate towards versus the others away? And this took me a while, and I went back in evolution a little bit along the way, sort of criticizing the tendency to outsource this question to reinforcement learning studies and saying, no, we can't do that because in reinforcement learning studies, like really in, conditioning studies, we condition behavior using something the animal already cares about, which means that, studies of the learning and performance of conditioned behavior can't tell us why the animal cares about that thing in particular. So eventually, I read more books and papers and got all the way back towards, the beginning of bilaterian brain evolution and found that there was something very understandable and interpretable going on with taxis navigation. Jordan, to tie you in a little bit here, there was that one time in energetics group where I sort of had this aha moment about a certain mathematical thing that you could do with like the vector of the direction you're going in interoceptive space versus the like another vector that you could measure in a certain way. And like, that came up again a couple years later when, as we're going to see, it turns out that like these combinations of vectors are things that people have found cells in some organisms that actually measure that kind of quantity exactly. So that came up again.
Cool.
So then here's my little hypothesis that I put down with Maxwell Ramstead in like a very free-form paper, basically saying once you evolve the ability to navigate a physical environment in such a way that you're coupling your internal states and your behavior to meet your allostatic needs. So once you evolve this ability to steer, then as the nervous system sort of, I believe it's as the nerve cord actually folds from bilaterians to chordates and vertebrates, this steering system can be reoriented towards your internal physiology entirely. So you can exapt and elaborate something that evolved to solve a very literally spatial problem to solve a metaphorically spatial problem while still using many of the same sensory and neurocomputational strategies. So I made myself some mathematical modeling. This is work in progress, actually, because, even last week I sort of jotted down some new notes on how that's going to work. But as for the biology, as I said, the actual like steering neurons that measure these sort of products of vectors, basically that measure the strength of a gradient in the direction the animal is facing, have literally been found in, the model nematode like the model nematode C. elegans. They have been found, their like extensive recording has been done from them, and we have a mathematical model that matches up exactly to what we would expect. So like there are specific cells, you can record from them, and their activity tracks the time derivative of a logarithm of a spatial density. And they actually receive, modulatory input via neuropeptides from interoceptive cells in the gut. So what I'm currently working on is basically like putting together and formalizing a very detailed model of that, and then aiming to show that when you move from bilaterians to vertebrates, we can understand this transition in terms of, navigating an internal physiological space rather than a literal environmental space. We can then try to understand, some of the neuroanatomy below the forebrain that, sort of the existing theory of constructed emotion sort of treats as just more sensory and motor effectors. Yeah. And we can try to sort of elaborate the theory from the beginning of evolution forwards as a way of getting at what kinds of computational models or neurocomputational strategies might be best used, rather than sort of starting from what I guess I would say is the cortex heavy, human behavior in the scanner, and then sort of developing theoretical principles from that, which are then applied backward in evolution. Yeah. So to give, a little elaboration to what I've been saying, this is a diagram from, I believe,
Physics
Ultransec
2019. Yeah.
2019. Yeah. Resynthesizing behavior through phylogenetic refinement. And he's basically like trying to say, well, what was the eventual evolutionary origin of reinforcement learning? Which I think at that time he sort of thought was the thing, followed through evolution by the development of species specific or relatively species specific or lineage specific, really, lineage specific neural circuits with distinct behavioral functions. And I'm trying to sort of go back and understand these bits where, how do you get something where, I'm sorry, let's see, it's not autonomic nervous system. Is it anterior nervous system and blastoporal nervous system?
Uh, yeah, ANS and BNS.
Yeah.
Yeah, yeah, yeah.
Basically, how did the evolutionary transition actually take place where this ANS and BNS are sort of elaborated into what Paul at one point claimed was, and I quote, the hypothalamus and the rest of the brain, which, I think that might be an overly strong statement, but certainly I think we could talk about, you know, like a C. elegans style exapted steering system plus a spinal cord plus a midbrain plus a forebrain and eventually get somewhere. Yeah. And that's actually about it for slides I had prepared.
Super. Thank you very much, guys, both. This is extremely interesting. In the time we have, could I ask a couple of questions?
For
So
sure.
yeah, so a few things. I'll start with the last thing first. This what I understood as this idea that the ability to steer around in space was eventually reoriented to steering internal physiology. So we've been studying this exactly a proposal of exactly backwards. So we do a lot of things on a lot of work on things that don't have a brain. So we're talking about single cells, various embryos, organisms without brains, and so on. And so what we see is biology navigating all kinds of spaces long before it had the ability to navigate three-dimensional space, right? So transcriptional space, physiological state space, anatomical morphous space during development, and things like that. I mean, we import a lot of things from neuroscience, these kinds of things of memories and making predictions and learning and errors and all of that. But like we see this in other spaces. So I've been proposing the opposite thing, which is that very early on it learned to navigate those spaces. Then it got some muscle and some nerve, and then it began to run around in 3D space. So that's
So if I could nuance my view there a little bit, I wasn't actually describing 3D space. I was thinking more like aqueous space. So like it's a physical space, but there's plenty of chemical signals to navigate by. So it would have been an instance of basically, multicellular organisms with very early brains just recapitulating, the behavioral strategies that were already used by single cell organisms immersed in their environment.
Yeah, I get it. Um, an interesting model system. Sorry, Jordan, did you want to say something?
Oh no, I was just going to jump in to say actually had the same question for Eli too. I'd written it down there that it could be flipped the other way as well, right? Because one of the books we've been reading as a group is Catherine Nave's Drive to Survive, which is really sort of an enactivist, like a biological enactivist perspective account of like digging into free energy and predictive coding and stuff. And one of the things I've been making notes on with that is that she she is interested in what the sort of like minimal normative push is that an organism can have for why it could want one thing instead of another or why it could be driven in some way. And she's coming from like the Varela like autopoietic perspective or like biological autonomy, and she's just trying to say that at a minimum, an organism needs to basically preserve its boundaries and replenish its parts, right? And so what she's trying to do is to say that that has to be the ultimate end that like any living system is working towards is to preserving its biological autonomy, and that navigating toward that, right, is what everything else has to be working towards. And so I was just curious too, yeah, if there's ways of navigating toward that or maintaining, like Mike was saying, like a transcriptomic space or something internal to itself, that that minimal normativity can be met by some internal navigation, which then gets projected externally once it has to suck in outside resources to persist for even longer, right?
Yeah. Yeah, very interesting. And, another model system that you guys might be interested in is we some years back wanted to ask the question, like, okay, there's a lot of development and morphogenesis that takes place before there's a brain, and we wanted to know how much of it relies on the brain, right? It can't be all of it, we know that, but like how much, right? So in the one model system that we work on is the tadpoles of Xenopus laevis. So these are frog, amphibian embryos, and we can make these critters without any brain at all. And so you can remove the brain early on. The rest of the body is completely fine. There are a couple of subtle things. There's something subtle that happens to their tail and a couple of other things that we actually have to work pretty hard to notice. But the rest of it actually works quite well. And they continue to develop. They become tadpoles. What they don't have is active behavior, so they won't run around and do interesting things. But they can. But they make it to pretty late stages, and they're fine. And so I'd be curious what, if any, predictions you would have about that kind of model. Like, one thing I wonder is, would you think then that without the brain that they would be using up more energy than otherwise? Because that's very testable if that's the case. What do you think? What, like, if we, I mean, I know this is hard in mammals, obviously, but in some of these organisms, if you could make one that just doesn't have a brain at all, what consequences would you foresee for that?
Just to, get the jump here, I think I would expect difficulties in motility. Based on the same old Carl Friston anecdote about the sponge, right?
Yeah.
Like if you make an organism with no brain, it's going to have difficulties at like moving in its environment to self-regulate as a motile organism.
I mean, it's true of those animals because there's no real muscle motion without the brain. They kind of actually, if you tap them, they will sort of come up and do this, but they don't have any initiative. They won't get up and sort of swim on their own much. Although we do have anthrobots and xenobots, which are multicellular organisms that absolutely navigate their environment and swim around. They don't have any neurons at all. I mean, it is hard, right? Because without interesting behavior, it's hard to do much. But on the other hand, if the idea is that the brain is important for regulating the internal milieu besides the brain, besides motile behavior,
Yeah.
then presumably there's something we should be able to notice about these things that's off. Something should
Yeah,
be
yeah, yeah.
internally, right?
That's interesting because yeah, I guess yeah, motility should be down like Eli said, but I guess I don't know if you could have a clear it it's hard for me to think of how to cash it out in the energy terms because part of the energetic efficiency should be energetically efficient motility, basically. But there should be something, I think part of the idea of an allostatic perspective, right, is that there's some coordinating, like a conductor function of a brain to coordinating all of those internal organs. So I wonder if you'd have something of if you would see some sort of coordination to challenge that you would see under other circumstances, like, even if it's like say you were exposing it to like a an environment of like intense heat or heat outside of a range that it should normally be in, do you normally see some sort of coordinated organ function to counteract that challenge, right? And do you see some sort of response of individual organ systems, but nothing in a coordinated way that helps it maintain that allostatic state, right? Like, it's tough because the fact that it doesn't move, there's a lot of stuff going on, but I'm trying to branch apart that, yeah, the two parts of the allostatic thing that we're talking about are both, like Eli said, I totally agree. Eli, when you were saying that there's maybe a difference of opinion at the start, I don't think there's a difference of opinion. I would say that the brain is for motor command first, motor control for regulating internal organs. Like you'd said in a
Like I
meeting a while
said,
ago,
I
we're
would
motor
really
chauvinists. Yeah.
I might just say that there's a difference of opinion if I'm in public to people who,
Sure.
to people for whom category construction really has a very narrow meaning.
For sure. But the point is that the category construction is also a means to the end. And the category construction is for efficient motor action through an environment, right? But
Okay.
oh no, sorry. The point I just wanted to make was that yeah, there's two horns of the problem. One is navigating an environment to get metabolically necessary resources. The other part is basically conducting and coordinating behavior of the internal viscera, which is, I think, more testable, like in the model that you're talking about there. If you could think about how to issue a challenge where you know what it should look like in a brained tadpole.
I think that's doable. Yeah, I mean, I was thinking of you had this very nice slide with going down to all the different organs, right? The muscle is one, but then there's all this other stuff. So
Yep.
the other stuff should be right. There should be predictions on that. So yeah, I
Yeah.
mean, sure, temperature regulation is a good one. I like it. We could test that.
Um, let's see. So I really do have to go, but before I do go, I want to make this suggestion. And Benjamin, I don't know if you could maybe, economics, right? So maybe you could help translate this into human words. I would wonder if you take an organism that normally has a brain, develop it with no brain, do its does its peripheral physiology suffer from some form of the bullwhip effect seen in economics?
That's an interesting question. So, my perspective is that the brain is analogous to a financial system. And so it's, kind of similar to a conductor in some ways, but a little bit different. Yeah, I see what you're saying about the bullwhip effect. I would expect that. I do actually have a blog post talking about how, like, Parkinson's disease might be analogous to a recession. So I think basically not having a brain or damage to the brain should be analogous to the problems we see when the financial system is in some way harmed or limited. Unfortunately, we don't have any
Yeah,
sizable
or
economies that don't have a financial system. Of course, that'd be very hard to develop, and we can't
yeah.
test that directly. But
For Jordan and Michael, before I go, the bullwhip effect is sort of this thing in supply chains where essentially if you don't have tight coordination between the different firms in your supply chains, then unpredicted changes in supply and demand at either end can suffer from positive feedback. So a small error, a small prediction error at the retail end becomes a larger prediction error at wholesale, becomes an even larger prediction error, in production,
I get it. I get it.
like at core industrial production. which then feeds in, which then the error really only attenuates when you get so far back in the supply chain that you're talking about raw inputs that are always bought and sold in monumental bulk. And so the typical solution to the bullwhip effect in, modern markets and modern firms is basically to have the firms tightly coordinate with each other to the point of usually sharing their databases and information systems. So if you look at like Amazon or Walmart, every retail sale is logged. And so when they get a prediction, or, essentially logged and to some degree predicted, and so they are sharing the data all the way back to the manufacturers in real time so that the manufacturer knows about consumer demand in real time.